drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to share with you a new AI-powered research tool I've been using to find scientific papers and learn from the literature. It's called Consensus. It is an AI-powered search tool built using large language models trained to help you find and understand the best science faster. Consensus helps you not only find individual papers, but its AI models will help you assess research quality and even synthesize insights across multiple papers for you. You can ask it general questions about any topic you might have, such as what are the known risks and benefits of a certain prescription drug? What do we know about how a certain food or nutrient affects the human body? Anything you can think of. Its Pro Analysis feature will actually provide you with a concise written summary of what the literature says, broken down point by point, highlighting the strength of the evidence across papers and providing you with individual studies ranked by the relevance and including additional information like citation rates, journal quality, and more. You can also save, cite, and share individual studies with a single click. Unlike other AI and search tools like ChatGPT, PubMed, or Google Scholar, Consensus uses over 20 custom large language models that are developed and trained for this specific purpose. The base Consensus product is totally free, but the premium product that you can subscribe to has better features and unlimited queries. And I highly recommend this if you are using outdated tools like PubMed or Google Scholar, or you just do your own research. For a limited time, you can use the code MINDMATTERSPECIAL, all one word, to get a free premium subscription for 1 year. That is a $150 value for free if you use the code MINDMATTERSPECIAL today. So click the link in the episode description or check my support page and give Consensus a try. Use the code MINDMATTERSPECIAL to unlock your free premium subscription today. Take a moment to each introduce yourselves and talk about what you guys study.
Sure. I'm Tim Yeatman. I'm a surgical oncologist by training. I'm currently a professor of surgery at the University of South Florida, and I'm an associate center director for translational research and clinical innovation at the Tampa General Cancer Institute, Tampa General Hospital Cancer Institute.
Ganesh Halade. I'm associate professor in the Department of Internal Medicine, a division of cardiovascular disease, and my research is more focused on inflammation and resolution signaling that focuses on the chronic inflammation that intersects not only cardiovascular disease, but also the cancer and other lifestyle-related disease.
Mm-hmm. And you guys have a really interesting new paper out. I read it just because it's the type of thing that comes into my my alert box. I've been talking to a lot of people on the podcast and reading on my own about topics that connect things like inflammation and diet to various chronic disease states, including cancer. And we're going to talk about lipids and diet stuff. We're going to talk about inflammation, and we're going to dive into your new paper. But there's also some background I want to get into at different levels. I want to give people a sense of how you guys think about chronic inflammation and factors that feed into it and cause it, including diet, including dietary lipids in particular. I want to talk about cancer and how you guys think about it and how the field has thought about it historically. And one place I thought was interesting to start, because I'm certainly not a historian, but I do love the history of science. You mentioned at the beginning of the new paper a man named Virchow. So who was this, and what were his ideas about what cancer actually is?
So maybe I can address that. I mean, Rudolf Virchow was I think he was born in 1821 and lived to 1902. He was a pioneering German physician and pathologist, and he's often regarded as the father of modern pathology. Basically, introduced the concept of cellular pathology, where he looked under the microscope at various tissues and tumors, and it's been attributed to him that he saw first inflammation in cancer, and inflammation was the influx of white cells basically into a tumor, immune white cells, and I think it's been also attributed to him and others that the concept of cancer is like a wound that won't heal. And I always remember you've had animals like a dog where they have sunspots, and that wound just won't heal. They keep licking, won't heal. It's sort of an ulcerated wound that won't heal. In a human, it might be a diabetic ulcer that won't heal, or an infected wound. And so he saw that, and remarkably, in the 1800s, identified the inflammatory process as being a component of cancer.
Yes. So the cancers, the virtue actually started the concept like how the inflammation is a fundamental there in context of developing towards kind of continuous process and uncontrolled. In reality, if we see the inflammation is designed helpful, but when it is in the acute process, and that is coordinated by the 1% of the immune cells that we are kind of having since kind of long time of history of the human beings. And that's kind of real historical perspective that how our immune system is built. And in that one, the Virchow added the kind of real key that how this chronic inflammation is involved, not only the acute inflammation, not only the kind of various type of metabolic diseases, but also in the cancer and many other diseases.
So is an idea here that inflammation, sort of on the one hand, we want inflammation. We want our bodies to be capable of this. It's a part of clearing out infections. It's a part of clearing out debris and tissue debris when we get physical damage. But I think, a key thing, at least for me and my understanding of inflammation, is you want it to be very spatiotemporally regulated. You want the inflammation to turn on at the appropriate time and then turn off at the appropriate time. You want it to be at the site of an infection or at the site of damage, but not anywhere else. You don't want it to bleed out to other tissues, because there's always this chance of collateral damage. And I don't know much about cancer biology in particular, or the history here, but it sounds like this idea that maybe goes back to Virchow is that if the inflammatory process itself becomes dysregulated, that can actually be a factor in causing cancer.
Yeah, so let's just go back a step. So evolutionarily, you might say that inflammation evolved as an important component of human existence and mammalian existence, because the greatest threat to life early on was probably infection, cuts and viral infections and bacterial infections. So we evolved to try to fight those things off. So inflammation, the whole body is geared towards turning on inflammation for that reason. I think evolution, it just kind of makes sense, right? That you'd really want to be ready to attack anything that comes at you. But you're right. If that inflammation persists inadvertently, can it cause problems? And I'll give you some great examples in cancer. So it's well known that inflammatory bowel disease, and that's ulcerative colitis and possibly Crohn's, but ulcerative colitis is a great example. In that disease, you see what's called leaky gut. And I always thought leaky gut was kind of a funky term that no one really understood, but the truth be told, it refers to the epithelial cells that normally have tight junctions, like your fingers sticking together, and they open up. And when they open up, they can release basically blood cells, lymphocytes, blood cells, neutrophils into the bowel lumen, which is the stool. So it's interesting that the most effective diagnostic test for active UC is something called calprotectin, stool calprotectin. Calprotectin is a neutrophil-specific marker. So if you see that in the stool, that means you've got neutrophils leaking from your capillaries into the stool. And that's why people with ulcerative colitis have bloody diarrhea.
I
So
see.
they now you understand that's what true leaky gut is, right?
Hmm.
And when you leak that gut, then you can have bacteria going both ways. And we should get a discussion of microbiome in the future, but let's put on pause for right now.
Hmm.
So you get this leaky gut and this inflammation, and it doesn't seem to go away much. Although in ulcerative colitis, it's called relapsing and remitting
Yeah.
because it's sort of you get inflammation, then it sort of heals. You get inflammation, it sort of heals. So there may actually be an attempt at resolution of inflammation in that disease. But if it goes on for long enough, inflammation, then you can get cancer. So it's well known that in ulcerative colitis, let go for 10 years, you increase your risk for cancer. That's why people with ulcerative colitis have to undergo uh, surveillance colonoscopy, and they get biopsies every 10 centimeters throughout their colon when they have this long-lasting inflammation, because there
Hmm.
is a risk. The other disease would be Barrett's esophagitis, a risk for esophageal cancer, some skin ulcers, cancer. So it's this sort of this chronic ulcerative condition that can lead to cancer. And ulceration, by the way, is a bad sign for all cancers. If you take melanoma, an ulcerated melanoma has a worse prognosis than a non-ulcerated one.
Hmm.
And why? Well, one theory I've got, and it's interesting. We've always thought of cancer as a true genetic disease, and it
Right.
is, right?
Right.
And from Bert Vogelstein and his group identifying APC, as the sort of the initial seminal mutation in many of these cancers, and subsequently getting KRAS and P53 mutations and other things like that. Um, we now know that maybe metabolism is part of this too. So
Hmm.
could cancer be a metabolic disorder as well as a genetic disorder? That's my question, and I think the answer is yes. So we now know that inflammation appears to be present in a tumor microenvironment, and that inflammation likely causes immunosuppression and reduced immunosurveillance. So
Mm.
one possibility would be that when you develop these common mutations in your billions of cells lining the colon wall, and that happens every day, and
Hmm.
they're normally destroyed by
Yeah.
your immune system. They become apoptotic and they go away. But what if you have an immunosuppression locally, a chronic immunosuppression? Would more of those take hold or take foot? And one example of that is a mouse study, and there's been a number of them, but there's a mouse model called the APC min mouse. And in
Hmm.
that mouse model, they basically have a single hit. In APC can have two hits, but one hit in the APC allele, and the mouse is born with that in every cell of its body. And if you give that mouse a highly obesogenic diet, rich in arachidonic acid, rich in omega-6 effectively, they produce many more polyps, 60% more
Hmm.
polyps than a normal mouse. So you're putting that inflammatory milieu in the presence of a mutation
Yeah.
that could cause cancer. And all of a sudden
Yeah.
you're getting more of them, more
Yeah.
hits.
Double
To
whammy, kind
me,
of.
to me, that's kind of de facto evidence that an inflamed environment could lead to increase, I don't know what is increased mutability, but increased chance of mutations taking foot.
Yeah. So if I'm tracking here, because I was actually about to ask you, as someone who's never been a cancer biologist, I never studied cancer biology per se, but as someone who's always in biology, I remember as a student sort of just having the notion in my mind, oh, cancer is from mutation. You get X number of mutations in tumor suppressor genes or whatever, and then the normal cell becomes a cancer cell, and off you go. If I'm following you, Tim, what you're basically saying is, yes, there are these mutations involved in creating cancer cells, but normally, I mean, there's so many cells in the body getting so many mutations every single day that all of us at some rate is generating, cancer cells. And oftentimes, more often than not, when things are working appropriately, our body takes care of that. Those cells are detected and they are destroyed, and the immune system is involved in that. It sounds like what you're saying, Tim, is that if you're in a certain inflammatory state, if you have chronic inflammation or what have you, then immune surveillance can actually be sort of turned down such that the probability that any one of those cancer cells that would have very likely been destroyed before it became full-blown cancer and problematic actually doesn't get destroyed. And so even though mutations are necessary to generate a cancer cell, the inflammatory state of the tissue can also dictate sort of the probability of whether or not that cancer cell will actually be handled when it should be.
Right, and we call that tumorigenesis, by the way. So, or carcinogenesis or tumorigenesis, basically the increased ability of the metabolic state to enhance tumorigenesis is what we think could be happening. It's also possible that once you have a tumor, that the inflammation can make things worse. And we don't know whether there's a threshold of inflammation or whether, in other words, you just exceed this threshold of inflammation, it's all bad, or whether there's a sort of a linear or logarithmic relationship between inflammation and immunosuppression. We do know that many of the cells in the tumor environment are macrophages, I think about 50%. And many of those are immunosuppressive macrophages. And we know that the card-carrying currency of the TME is lipid mediators. And so having a lot of these inflammatory lipid mediators in the tumor microenvironment likely contributes to this immunosuppression and decreased immunosurveillance.
And when you say lipid mediators, is the basic idea here that you've got all of these different immune cells. Some of them can stimulate inflammation, some of them can suppress the immune system. When you say lipid mediators, you mean that these are molecules that immune cells have or they release that are sort of the information signals that are saying turn inflammation up or down and things like that?
I'm going to let Dr. Halade, the expert on, take you maybe through the pathway from maybe a seed oil that has linoleic acid, to arachidonic acid, to how you get to this these lipid mediators and what they actually are.
Yeah.
Sure. Yeah, I like to kind of glad. Before that, I just want to use the analogy for the chronic inflammation. Like, what happened when you have acute inflammation. It's just like, when there is a fire, firefighters come very precisely on time, control the fire, and they leave. But in the chronic inflammation, the first thing is these firefighters don't come on time. And even if they come on, they just hang on where there is a fire and they forget to leave. So the immune cells get in signal, their transit signal and get out signal is totally discoordinated. And that's where the Tim Yeatman's really nicely kind of explain how the immune suppression is developing. So that's the discoordination of that leukocytes that's developing the chronic inflammation. That is kind of where many metabolic diseases are kind of developing the whole process.
I see. So you have signals calling in the firefighters, separate signals telling them when to leave. And if these things aren't properly coordinated, the firefighters go to the wrong place at the wrong time.
Wrong time, exactly. And plus, especially if they are coming late at the tumor site, plus they are hanging, doing the party with the fats that every day we are taking in the body, and then just there is a continuous progression of the tumor, and that's whole tumor environment we have described in the paper. That's now here is the question of how these molecules are formed. And we know that historically that these fatty acids, they are not synthesized in our body. That's why they called as essential fatty acids. And then those are necessary 1 to 3 to 5%, depending on where we are on the growth curve in our life. Like if we are like early in the adult phase, we need slightly kind of more quantity. And then down the line, as I'm old or, in the kind of late phase life, we may need relatively lower. But what happened to these fats or essential fatty acids? They get used by the immune cells more rapidly in response to acute inflammation and make number of lipid mediators. Same process happen in the chronic inflammation where these fatty acids used by those discoordinated firefighters or the immune cells, where these fatty acids from the diet get converted into the second generation of those molecules. And those second generation molecules we called as lipid mediators or the bioactive lipids. And here we need to note this point that these second generation molecules are more potent than the actual original fatty acids present in the diet. And as you know, and that the standard American diet is enriched with the linoleic acid. That linoleic acid get converted into arachidonic acid, and that further forms the many second generation molecules, and those are the inviting the immune cells. That is the first step of the initiation of the chronic inflammation. So that's how these lipid mediators forms a continuous process that invites more and more immune cells, that leads to the tumor progression and kind of continuous growth of the tumor in various regions of body depending on where it is initiating. That is one step. Now, in contrast to that, if you think about, imagine instead of this unhealthy fat, you have the healthy fat, which is coming from omega-3 and other kind of similar related sources, fish or other related sources. And then the same enzymatic system and the immune cell converts those to the healthy molecules. Those are called as a resolution mediators, and that helps to control the immune cells or give the immune cells to get out signals, and there is a clearance of the system and limit the inflammation. So there are totally two arms of the process in the body. Imagine if we have unhealthy fat, then our enzyme system with the help of immune cells, then that unhealthy continuously kind of feeds the chronic inflammation. In contrast, if we have a healthy fat, then our immune system with the help of this immune responsive metabolic enzymes make the molecules, helps to clear the system. So that's how this process, if it is really balanced, then we stay healthy, but if it is imbalanced, then leads to continuous chronic inflammation. But there is a very tight coordination of the fat, what we take every day, and the enzymes present in the immune cells, especially the leukocytes, that determines the biosynthesis of those lipid mediators. And down the line, those lipid mediators, those are very potent than the fatty acids, actual fat in the diet, and gives the clear signal to our immune cells.
Mm-hmm. So we've got these lipid mediators that are super important for the inflammatory response, how it's going to initiate, how it's going to resolve. And in essence, it seems like there needs to be a balance of sort of two classes here, the pro-inflammation mediators and then the resolving mediators. And these have to do with things like dietary fatty acids. So we eat fat. Certain fatty acids serve as precursors to these different lipid mediators. One thing I want to talk about here is this concept of balance versus imbalance that's going to become important, I think. So is it true that if I just eat a bunch of fat, let's just say I'm not tracking it too closely. That's probably true for a lot of people. I'm not paying too much attention to the specific fatty acids I'm eating. Seed oils, saturated fat, omega-3s, you name it. I'm just sort of eating what I eat. Can't my body doesn't it just know how to make the perfect needed balance of these things? Or does the balance of what's coming into me from my diet really dictate whether or not there's going to be an imbalance?
So have you ever heard that phrase, you are what you eat?
Yeah.
I think there's some truth to that. So we have to, again, get into the biology of this a bit, but the enzymes that process omega-6 derived fatty acids or omega-3, they're actually the same. The ones that process omega-3 and 6 are the same enzymes. So it's called ALOX5 and 5-AP. And those enzymes can handle either one, the pro-inflammatory or the pro-resolving lipid precursor, omega-6 or omega-3. So as with any enzyme, if you give it a substrate in excess, it's going to favor that substrate if it's got equal propensity to affect both.
I see.
And so if you load your body up with omega-6s at a ratio of 30-1 or 25-1, your enzyme is going to be more likely to convert that to a pro-inflammatory lipid mediator than a pro-resolving lipid mediator. And that's kind of, we think, what happens where we have, our diet is so over overwhelmed with omega-6s. You can't eat enough omega-3s to balance it. The lipid mediators are evanescent, by the way. They get turned on and off, like you said. It's a temporal thing. It's timed with inflammation. And there's a certain time when these things come on. There's a certain time when they should go off. But the substrates are always floating around, right? And so there's a nuance to the science here, understanding what the substrates are and then what the lipid mediators are, and measuring both. So if you look at, and I looked at myself, I have a 13-to-1 ratio, for example. Ideally, it'd be 1-to-1 or 2-to-1, favoring, more towards the omega-3. You'd like two omega-6 to 1 omega-3.
Yeah.
But like I'm 13-to-1, but the average American may be 25-to-1 or 20-to-1. Now, if you look at the foods we eat, let's take beef, for example. Beef or pork or anything of that nature, it's about 25 to 30-to-1 because the beef is grain fed. And you've heard of fattening the calf with grain, using grain to fat. Well, that's why it's basically this sort of carbohydrate rich diet and there may be, by the way, maybe soybean oil in things like cattle feed. I don't know for a fact. But if you're getting a grain fed animal, that's where the meat is now 25-to-1. And you're getting every bread that you eat, or every cookie or piece of pie or whatever you're eating is loaded with, basically omega-6s, then your body's going to see much more of that. So you are what you eat. I think you're going to be flooding your receptors and your enzymes with this inappropriate ratio.
Yeah.
Now, what can you do to correct that? You can, well, like if you go to grass-fed beef, the ratio is 1-to-1.
Yeah.
Amazingly. And grass-fed milk, 1-to-1, and grass-fed butter, 1-to-1. So just switching to grass-fed, animals and milk can really help, because many people eat a lot of meat and butter and milk, and so that's one thing you can do. The other thing you can do is try to be vigilant, looking at labels and seeing what you're actually eating. But you have to have a microscope to get the labels. When you see so many ingredients, it's hard to actually find where the soybean oil is, but you can see it if you look very carefully.
Yeah. Well, I mean, I think one of the places, one of the things implied here is, it's not sufficient just to look at the total fat content. You've got to actually go down at least one more level and say how much is polyunsaturated, how much is saturated, and so forth.
There's a lot of literature. We grew up, I would say, in the early days where it was butter and Crisco. My grandmother was known to have made a jingle for Crisco: north, south, east, west, creamy Crisco is the best. And of course, we got away from all that because we realized that saturated fats were bad too in excess, right? And they can cause heart disease. And then I think when people moved over to margarine and things like that, maybe that helped heart disease to some degree. But when you go full spectrum and put every food you have loaded with these types of oils, then you've changed the balance. So you may have reduced the saturated fats, which is good, but you've increased the polyunsaturated to the inflammatory side, which can be bad. So again, life is a balance, and eat in moderation is the right approach. Do everything in moderation.
Yeah. And so I want to get into the idea of balance more and really dig into some of the biochemistry before getting to the paper. But just to tie off on some terminology, because we're going to have people listening with variable levels of knowledge coming into this. So when you talk about, I want to sort of make it clear for people, what are omega-3s? What are omega-6s? When you guys talk about omega-6s, you're talking about certain polyunsaturated fatty acids that come to us from our diet. And Tim, a number of the things that you said, and I'll let you guys expand on this, but a number of the things that you said are the average American, the average Westerner, probably the average person on planet Earth at this point, is getting an imbalance of omega-6s and omega-3s. So we're getting way more omega-6s, most of us, compared to omega-3s. Historically, in evolutionary terms, if we go back before industrialization, back in time, most people were consuming a pretty balanced ratio of these omega-3s and omega-6s. And these omega-6s are what people refer to as either vegetable oils or seed oils. Is that correct?
Yes, that's correct.
That's correct. So what happened is kind of now our source of the kind of food intake is slightly changed. We moved from home cooked food to the processed and packaged food. So that's the source. Then from there, why the processed food industry will use the healthy oil for your health. So the majority, they use the seed oils, which are much more cheaper, so that they can make more revenue, but that is costing to our health because of the presence. Now, for that one, we will use the easiest way, the car analogy. So what happens if our car oil goes bad? We go and take out the old oil and put the new oil. But here in the human, we don't have that knob to take out the old oil. And even if we go switch to another healthy, but that old oil is still there a long time in the body. This is very simplified version of this. And for the work the car perfect manner, need a kind of sort of four wheels. Same way happens into the fat. So the fat is the number one component that we take from the environment, from different products. Then that get used by the enzymes which is present in body. And then the third product is the lipid mediators. And once these lipid mediators are formed, then the last wheel is the receptor. So the diet, enzymes, lipid mediator, and receptors, those all are the four wheels that makes or decides whether the fat is going to either helpful or harmful side. Now, the first step itself, our body is fully loaded with the omega-6 continuously from the environment, then obviously it's driving towards the pro-inflammatory side. Obviously, it has no choice that the enzymes using that because of the abundance, making more pro-inflammatory molecules, and the receptor related to the pro-inflammatory molecules, those are constantly activated. So the body is kind of going towards the pro-inflammatory. This is kind of super simplified version of very complex pathway, because one single fatty acids like omega-3 linoleic acid gets to the arachidonic acid. The arachidonic acid makes more than 20 to 25 molecules at the site of tumor or at the site of injury, depending on the progression. So that's how the fat biology, even it's very complex, but this is kind of very simplified version that how the imbalance of the omega-6 to omega-3 is kind of leads to that point of the chronic inflammation because of these key four components driving or feeding the chronic inflammation.
Yeah.
So I wanted.
Oh, go ahead, Tim.
I was going to add to that you can look at omega-6 to omega-3 ratios in body fat, and over the years from 1950 to more recently, there's been a tremendous rise in the omega-6 to omega-3 ratio in the Western population, and so that's prima facie evidence that you're taking more in, and the diets have changed.
Yeah,
and so that's a fact. And the other thing that's interesting is fat, when you eat fat, it can go everywhere in your body, basically. So there's a guy named Garth Nicholson who identified the lipid bilayer of the cell membrane is two lipid bilayers, right? So or two layers, a lipid bilayer, and phospholipids, right, compose this the cell wall of all our cells. So those fats that you're eating get inserted into every cell in your body.
Yeah.
And so over a course of decades of eating, the wrong diet, you can imagine that all your cell walls have that makeup, right? That constituency. The other thing that's interesting is there is a enzyme called phospholipase A2. And that's an enzyme that releases arachidonic acid from the lipid cell wall. And phospholipase A2 is actually a cardiac test for inflammation that you can actually get with LabCorp and Quest. You can measure your level of inflammation, and I think it's different than measuring C-reactive protein that a lot of people use. So it's interesting. How do you determine inflammation in the body? One is to look at phospholipase A2 levels, and they're active in releasing arachidonic acid.
And Tim, I want I'm gonna say here that I want to come back to that maybe because one of the things that often gets brought up in this realm among aficionados or people who claim to be is there's some maybe gaps in the clinical literature where people will look for things like CRP or certain inflammatory markers, and they'll say, well, we don't really see a correlation between intake of these things and inflammation. Inflammation meaning like the one marker they look for. I think what you're saying is you have to look for the right markers in the right places in order to pick things up properly.
Absolutely. And I think that, plasma levels of things are different than tissue levels of things.
Mm-hmm.
So people will look at plasma levels and say, well, I didn't see anything there. But you look at the tissue, it's a different story. So I think the science is still in its infancy in understanding lipidomics and lipids we eat. And it's remember, people are complex. And you also remember there's heterogeneity. So if you look at, one person, another person, even our own data, we saw some people with sort of very high LTB4 levels and some people with less high and some with lower levels. And you say, well, on par, they were all elevated. But why is the heterogeneity there? What does that mean? And again, I don't know yet. We're going to look into it with more samples. It's certainly possible there's a threshold that you exceed. Like I said before, where if you exceed a certain threshold of like LTB4, you're inflamed.
Mm-hmm.
Or it may be that a really high level is worse than a really low level. We don't know yet, but clearly there's an on par average median elevation of these things.
But okay, so let me see if I can tie some things together and maybe simplify a little bit and then dig into what if I'm hearing you guys correctly, some of the key biochemistry for why certain things are true. So you've got omega-6s, you've got omega-3s, you've got other types of fatty acids, but if we're just talking about the PUFAs, the polyunsaturated fatty acids, two big classes, omega-6s, those are what we refer to when we're talking about seed oils. Then you've got omega-3s, that's typically referred to as fish oil. Omega-6s are going to serve as the precursors to the pro-inflammatory lipid mediators most of the time. The omega-3s are basically going to serve as the precursors to the resolving lipid mediators. And so you want these things in balance. And it sounds like it really does matter what your balance is on the intake side, your fatty acid intake, the food you're eating and its fat content. Because when these things the reason these things go out of balance, I think ties to what you said, Tim, which is they have the same enzyme is responsible for processing both of them. And so there's sort of like this rate limiting thing or this funnel where if you've got an imbalance on the intake side, then you're going to produce more of the one class of lipid mediators on the output side of that enzyme because it's the same enzyme processing both the omega-6s and the omega-3s. And so unfortunately, it would be great if your body could just take an arbitrary set of fatty acids and then make exactly everything in perfect balance. But based on this biochemical limit here and how these enzymes work, that's not the case. And so your intake really needs to be balanced, if I'm hearing you guys correctly.
And supporting that, there are some studies that are very interesting. We insurance companies have used, LDL levels and say, get your LDL level below 100 and that's healthy.
Yeah.
Get your HDL level up a little bit, that's healthy. But I don't know whether anybody's ever used omega-6 to omega-3 ratios. And it might be a better index of health because there are studies that show that a higher omega-6 to omega-3 ratio in plasma is linked to increased mortality risk from all causes. Cancer, cardiovascular disease, individuals have the highest ratios and have had a 26% higher risk of mortality and a 14% higher cancer mortality. So and there's some confusion on this, some recent studies where some cancers show a lower incidence with higher ratios, like breast cancer, but it's small. It's like a decreased 8%. So a lot more work needs to be done here, but the concept that omega-6 to omega-3 ratio might be important indicator of health, I think is something that needs to be explored.
Mm-hmm. Mm-hmm. And go ahead, Ganesh, were you going to say something?
Yeah, I just want to point out that, as already Tim mentioned, you are what you eat, but again, there is slightly more than that there. So when you eat what you eat and how much you eat, so those are getting granular because down the line, your, everyday biology in context of circadian rhythm contributing to that component. And that's where it is coming to the point that when these lipid mediators form using the processed fat, and sometimes people think that, hey, those are absolutely kind of, these seed oils are bad. But one point we just need to understand their name. They called as essential fatty acids, means they are absolutely essential into 1 to 3% for the initiation of inflammation when there is injury or infection, because those molecules are the one gives a signal to our immune cells, hey, there is a problem, come over there. So
Yeah.
those invite the immune cells, that is there.
Yeah.
But here is the what happening is that they are continuously giving that signal, hey, come here at the site of the tumor, here party is going
Yeah.
on.
Ganesh,
So
I think you said that the omega 6s are necessary at 1 to 3%. What did you mean by that 1 to 3% of calories?
1 to 3% in terms of their, depending on the growth curve. If you are in the adult phase, then probably in the 1 to 3% in terms of the kind of how much you are taking as in the form of the grams, how much you are taking in the diet.
I see.
Yeah.
So grams. So a tiny percentage should be.
Yeah, tiny percentage, and that also needs to kind of club with how much you are active versus what's your sleeping pattern. So
Yes.
those all three components necessary for the activity of the enzymes machinery, what the team was mentioning, that because this machinery process both the omega-3 and omega-6 in response to injury.
Mm-hmm.
or the kind of tumor where there is a continuous chronic inflammation, where the lipid mediators are formed, that we have quantitated by using very state-of-the-art mass spectrometry.
Yeah, but it's. Oh, go ahead, Tim.
Well, I wanted to get back to what's healthy and what's not healthy. So we think a balanced ratio of omega-6 to omega-3 in your plasma, a balanced ratio in your food, therefore. Makes the most sense. It's close to 1 to 1 as possible. And again, the UK Biobank study of 86,000 patients found increases in all-cause mortality and cancer mortality and cardiovascular disease increased with the increased omega-6 to 3 ratio. So that's really good evidence. But if you go the opposite direction, let's say you really, remember, omega-6s are essential fatty acids. You have to have them to live.
Yeah.
But it's the question of how much, right?
Mm-hmm.
But if you go the opposite direction and try to reduce your ratio from 1 to 1 to 1 to 4, in other words, 4 on the omega 3 side and 1 on the omega-6 side, that's very hard to do. But apparently the Inuits of Alaska have that sort of ratio, and they have a high risk of heart disease. So if you imbalance it the other way, you may actually increase problems too. So it's this balance, this life balance that Dr. Halade often talks about. Is important.
Yeah. And so getting to the question, so we talked about sort of the biochemical reason why balance makes sense and 1 to 1 makes sense, because the omega-6s and omega-3s kind of get processed by the same enzyme. And in some sense, you might say that the body is expecting balanced input. And so I want to ask if that's true. And so if we think about this from an evolutionary perspective here, would you say it's accurate or reasonable to say that for most of human history, before we started industrialization, when we were, out in the wild, so to speak, except in extreme environments, perhaps like where the Inuit lived, Tim, that in most natural environments, humans we were intaking a 1 to 1 ratio of omega-6s to omega-3s because that was what was more or less naturally present in the environment. It was only after we started creating synthetic environments, industrial environments, or, in maybe extreme cases where some people moved up to the Arctic, where you got this imbalance of omega-6 and omega-3 inputs.
Yeah, you're absolutely right. So modern agriculture, big agriculture, has tried to mass produce foods at a lower cost.
Mm-hmm.
And so you're right. In those days, I'm sure that all beef was grown on grass and had this 1 to 1 ratio. And I hear that it takes about 3 years to bring a steer to market on grass-fed versus maybe 1 year on grain-fed. So there's an economic advantage to go with the grain feeding, and that's where we've gone. But the same thing's true with these other oils. It's a lot cheaper to put in canola oil, which is horrendous processing to get there from seed, if you actually read and look how it's actually processed, or soybean oil or cottonseed oil or sunflower oil versus an olive oil or avocado oil. Those are more expensive. So we've put these cheaper oils into things and tried to promote the idea that omega-6s are healthy, but in fact, just this imbalance has in everything now. So you can't go overboard, and we have in all our foodstuffs.
Yeah, yeah.
Yeah. One thing happened is that our kind of food source is changed, and that is shifting the omega-6 to omega-3. But at the same time, what happened? We have really kind of our whole lifestyle becomes more sedentary. So
Right.
we increase our pro-inflammatory input in the body, but same time we become sedentary. Another one confounding factor is added, that we are less sleeping. So that kind of what we may ask, like, okay, what is the connection of these two components? One, that is kind of because of the pro-inflammatory input, the body is kind of moving towards the obesity, and that kind of correlate to the kind of number of high cases of the cancer. And the component, the lesser, these both the components actually impacting the enzymes which processes the fat, whether it is omega-3 or omega-6. Then if your enzymes become weak because either overabundance or your sedentary lifestyle or you are not sleeping on time, that leads to obviously what happened, that whatever intake you are taking, your machinery to process the fat is already cramped, weak. So the quality of mediators will be a very fragile or totally collapse or disrupt. And that's why whatever you are eating, it's not getting cleared, or whatever the debris is from different sites in the body, that is not clear. That is the kind of primary function of the immune cells when you are sleeping, as a patrolling different immune cells, go in a different sites, find out the wear and tear and clear it. So that's how kind of hold the immune system and the fat, they have very tight coordination every single day, and find out where is the debris, how it can be cleared with the help of those four components: the fat, enzymes, lipid mediators, and the receptors. And they work as a very precise, tight fashion so that they can clean the body and maintain the health.
Yeah. And then so one of the themes here, I think, is that there are multiple points of variability here, or where the potential for inflammation can be altered. So one is just, what's the total amount of fatty acid substrate you have to work with to begin with, and that gets into this idea of the omega-6 omega-3 balance in terms of the fat component of our diet. But also, Ganesh, as you're saying, there are other factors that determine the extent to which true inflammation will come from that potential, and that ties into things like lifestyle. How much are you moving? How sedentary are you? How much stress is there? What are your sleep patterns like? All of that will tune the extent to which that inflammatory potential from the omega-6s actually then gets turned into bona fide inflammation in the body.
Nick, you're describing systems biology. So systems biology is the concept that you just can't look at one molecule, one cell in isolation. You have to understand how everything is orchestrated as a system, and that when you perturb the system, it's like the butterfly effect. Sometimes you can perturb many different things. And I've always thought that genetic engineering was interesting, right? I actually wrote a paper years ago, Egypt in the year 2000, and this was a paper written in 76 predicting the use of genetic engineering in agriculture. But the reality is that when we put a single gene into a cell, do we really know what effect that one protein produced by that gene, or knocking out a gene, is doing on the other 20,000 genes and 150,000 proteins? Are there subtle changes in patterns we don't know that can stimulate autoimmunity in the body, for example, and all of a sudden your body's recognizing a form of wheat that is not natural, in the sense that there was a genetic modification, and it didn't seem on the surface to have any toxicity or anything, but in fact your body is recognizing it as slightly different, because your gut is this incredible immune system, right? And so it has great immune potential and autoimmune potential. So I think we have to be more careful in the future about what we put into our mouths and what we put into our foods. I did want to mention one other thing too, is you were talking about the evolution of agriculture and food and everything. It dawned on me that a lot of these ultra processed foods are also present in the fast food restaurant change. And we've gone to a fast food mentality. And, whether you're getting, bagels with soybean oil in it or cookies or cakes or whatever. And I won't mention any particular companies, but they're everywhere, right?
They're
And they're every
everywhere, yeah.
corner. And so, but guess where they're often in every corner? Also in rural America,
Yeah.
very populated there. And it's interesting to see that rural America has what they call food deserts, and there is an overwhelming incidence of obesity in these areas
Hmm.
and cancer. So
Hmm.
we've recently mapped out in Florida the obesity zones and the rural America zones, and quite frankly the zones where there's a lot of ultra processed food. So there's an overlap. I'm not saying it's cause and effect, but
Yeah.
it certainly
Yeah.
is a smoking gun for that sort of thing.
Yeah.
So you
Yeah.
question whether, the health of this country is going the wrong direction because of the food we eat.
Hmm.
And of course, that's a very popular topic today with RFK Jr. talking about, making America healthy again and all that. But bottom line is, if we focused more on health, maybe reduce these chronic diseases that are taxing our Medicare dollars. I mean, most of our spend today in CMS is based on chronic diseases. It's diabetes and heart disease and heart failure. These are like the number one, and Alzheimer's is going up there. So these are all chronic inflamed diseases driving our healthcare costs. So are we completely focusing on the wrong things, right? We're focusing on disease treatment rather than disease prevention. So
Hmm.
one of my shticks, I would say, is that if we could focus the healthcare system more on preventing
Yeah.
disease
Yeah.
than treating it. And right now we only treat it, basically. Doctors are trained to treat, not prevent. That's important change we need to make.
Yeah. And I like what you said about the food deserts, Tim, because I think that language actually sort of betrays some problems I think that are just widespread in how people think about these things. That the concept of a food desert is really, in certain areas, in poor areas of America, people lack food with good nutrition. And sure, that is true, no dispute there. Like, if all you have is the 7-Eleven, there's a lot of things you don't have. There's a lot of vitamins you're not getting. But what you're saying is actually what we're missing there is, well, a lot of that unhealthiness might come from what they are getting at too concentrated levels. And so it's not just that you're lacking a few vitamins and minerals. It might be that a lot of that unhealthiness is driven by what's there too much.
Correct. I mean, and we don't see, the starvation of Biafra. Remember the years ago, we'd see these poor children with large bellies because they were swollen with edema. They weren't getting enough protein in their
Yeah.
bodies. We don't see that, but we see the different swelling of the abdomen from fat. And so you're right. The food desert is sort of a misnomer because there's plenty of food there. It's just the wrong type, and it's cheap food, and so consequently, people eat it.
Yeah.
Now, it's been said to me that if you started trying to change the food contents with these seed oils, replacing with olive oil or avocado oil or nut oil, walnuts and almond oil and things like that are all good. It would drive the cost of food up. But what have we done to the cost of healthcare? And what's the leading cost of the deficit today? It's healthcare and the Defense Department. So those are the two cost drivers. So if we're really going to focus on the future, we
Yeah.
really ought to focus on the health and the food we eat.
Yeah.
And that in the end may drive down CMS costs that are associated with this budget
Yeah.
problem.
Yeah. And so, I think we should dig into the paper now. It was a really interesting paper, and people are definitely talking about it. Well, actually, one more thing I want to ask by way of background is on the topic of variability and the subject of why, because we're talking about diet to some extent here. Obviously, everyone's not the same when it comes to how diets affect them. We all eat different things, but we're also differently affected, right? Some people are lactose tolerant, some are lactose intolerant, et cetera, et cetera, et cetera. One of the things, my understanding is that there's actually a lot of variability in fatty acid metabolism between people, and that includes the enzymes that process some of these omega-6 and omega-3 fatty acids, such that some people much more readily produce from the omega-6 dietary fatty acid inputs, things like arachidonic acid and the stuff you guys were talking about. Is that true? Is there a lot of variability in sort of our sensitivity to seed oils in effect?
Yes.
I'm going to Let
Go ahead.
Ganesh talk, and then I'll say something too.
Yeah. Okay. So variability, it's a very obvious and expected one because everybody is taking different products. So different products has a different content, different source, and different levels of fatty acid. That is one thing. But also the every individual have a different set of movements as a physical activity or work style that is impacting the activity of the enzyme and also the sleep. So those are the kind of very obvious, everyone is doing that. Those are the obvious compounding factors that changing the kind of density or activity of those omega-3 or omega-6 processing fatty acids. And as Tim already hit that, we have all over these all diseases everywhere, and whether it is cancer or the heart disease or the Alzheimer's, and they are kind of widespread. You know, Now the key question is they all related to the metabolism. Now we are just talking about the fat metabolism, whether it's applicable to the protein or other components, those are present in the different types of food. And because our body serves as a machinery with the help of enzymatic machinery that makes a number of molecules. So we're just kind of scratching the surface here that how this one particular type of fatty acids is making different molecules. But of course, the variability is coming from everyone's lifestyle related confounding factors that's driving changes in the enzymatic activity.
So I want to add to that when we looked at our tumors, the enzymes that are processing these bad lipids, ALOX5 and ALOX5AP and LTA4H, those enzymes were all increased in expression in the tumors versus the normal samples. So it's sort of a double whammy. Not only is there more substrate, but there's more of this enzyme activity in the tumors, again, contributing to this inflamed environment. So if you have more enzyme and more substrate, you're going to get more byproduct, which is the LTB4, the 5 heats, these other things that we saw.
Mm-hmm. And so, based on what I've heard here and sort of my read of your paper, which I want to dig through a little bit, it sounds like the impetus for this paper was, okay, colon cancer is a thing. It's a problem. Where does it come from? And obviously this ties into, I think, larger questions on just where cancer comes from more generally, but you guys were looking at colon cancer here. And then this, colon cancer in particular is probably related to diet more than other cancers because, our food goes through our intestinal lumen and there's a pretty clear connection there. But it seems like the idea here was, okay, if we think it's possible that lipid dysregulation might be an important part of cancer or colon cancer, and we know that we've got these pro-inflammatory lipid mediators, we've got these anti-inflammatory or pro-resolving lipid mediators. We know that dietary fatty acids influence how much of each of those you have. And we know a fair amount about some of the biochemistry here, and that's some of the stuff that you guys were telling us about. It seems like the hypothesis here is that, well, maybe people that develop colon cancer have a certain type of lipid dysregulation, and that is potentially coming from some kind of imbalance in their diet that's promoting this more pro-inflammatory state, and that ties into seed oils and all that stuff. Is that roughly what the thinking was going into this study, or how did you come to even do these experiments?
Yeah, so I can tell you, one of our senior scientists in the lab, Ramani Sanrajaran, she came to me one day, and when we were starting the lab, we just moved it from Utah, and we were starting up, and we were studying the genetics of colon cancer, and she said to me, Dr. Yeatman, there's this physician, this scientist down the hallway, Dr. Halade, who's studying lipidomics, and you really should consider, measuring some lipids in these tumors. And I was thinking, oh, okay, I'll get around to it, And she was persistent, I have to say. She probably told me 2 or 3 times, and I said, okay, let's send him over 10 samples and see what happens. And I really didn't know much about lipidomics then, and I didn't know how they were important in communicating with this tumor microenvironment. I didn't know any of that. But when I got the data back and it all showed sort of one thing in 10 tumors, I said, boy, that's interesting. What does it mean? So I then met with Dr. Halade and started doing my homework, and it literally took me 6 months to get a knowledge base, and I can show you the papers piled up behind me here, to read them, to understand the science that leads up to all this. So it was a little bit serendipitous the way it started, but now we've become laser focused on, these lipids. And there's another whole angle to this story, which is, you eat lipids and they go down your small intestine and they hit what? They hit some bacteria in the small intestine, probably in the proximal colon. And those bacteria do things to the lipids. They break them up. They may hydrolyze things. Not quite sure what they do yet. But there's the microbiome of the gut, which is another whole story within a story here.
Right.
And we know now that simple artificial sugars or like sucralose, for example, can dramatically affect your microbiome. And if you go to the grocery store again and look for labels, you'll see sucralose is in a lot of things. I personally don't tolerate it very well. I can tell when I've had it.
Mm-hmm.
And the question is, what is it doing to your microbiome? Well, people have done that study, and they've looked at sucralose and all sorts of other these sugar alcohols, erythritol and things. And they, in fact, do change the diversity of your microbiome. They lower the diversity. Diversity is considered something to be good for the microbiome. You want more different types of bacteria because there's an effect that's beneficial that way. So if these simple sugar substitutes are affecting the microbiome, what is everything else on that food label doing to it, including the lipids? So if you overload your body with omega-6s, how is the microbiome responding? I don't think we know that answer yet, but I think it probably does adapt. I think that certain bacteria thrive with certain types of lipids, just as they do with certain types of sugars. And that's yet to be really studied well. So it's another whole universe to study.
Yeah. So why don't we walk people through the study a little bit? Does one of you want to just give us the basic setup here in terms of what you were looking at it and sort of what experiments you were doing?
I can start and then Dr. Halade can finish. So we decided to look at about 81 tumors, but we thought it was important to pair those tumors with normal mucosal samples taken about 10 centimeters away from the tumor itself.
Within the same individual.
Right, same person. You, as a surgeon, you cut out a segment of bowel that may be 1 to 2 feet length, and the tumor is sort of at one end and normal mucosa at the other end. And so you make sure you're far enough away, there's no tumor nearby.
Yeah.
And we sent both of those, we froze them, snap froze them in liquid nitrogen within 15 minutes of removal, and carted them over to the laboratory in a de-identified fashion and analyzed those samples for a portfolio of lipids and lipid mediators.
Wow.
And that was the setup. Now, we also sent some to Japan to look at untargeted lipids. So we did shotgun lipidomics, looking for various lipids that might be overexpressed or underexpressed in tumor versus normal. And then we focused in Dr. Halade's lab, he's focused on the arachidonic acid pathway. We went to quantitative lipids. So in the untargeted, it wasn't quantitative, it was fold changes. In the Halade lab, the arachidonic acid pathway is all quantitative. So we're getting down to picograms, nanograms, picograms per milligram of tissue.
I see. So you really had a multi-pronged strategy here where you could sort of take a bird's eye view, the shotgun approach, and say, like, let's just see in general if anything seems to be really out of whack. But then also in Ganesh's lab, it's much more precision. You probably had candidate molecules you knew might be important here, and you could get really precise with the measurements. And then you've got not only 80 some odd individuals you're talking about, but as you said, Tim, you're actually taking multiple samples within individuals so that you're getting tumor tissue and healthy tissue for each person.
Right. And then exactly right. You nailed it. So the reason we took the normal too was because historically people have just looked at, and they've done this with gene expression profiling too. When I first got involved with gene expression profiling of tumors, we take tumor and normal, and you'd see, oh, these genes are really increasing in the tumor versus the normal. And then a lot of people started publishing just tumors alone. Mm-hmm. If you get enough tumor samples, you can look at tumors by themselves. But when you're doing smaller numbers, you really need to compare tumor to normal. And we saw significant differences in that really didn't match up with some of the literature, the older literature. And so the older literature used, for example, radioimmunoassays to detect prostaglandins. And we used lipid chromatography, tandem mass spec, much more sensitive, much more specific. And so when in the older literature, someone thinks they're looking at, one particular LTB4 or LTC4, LTD4, LTE4, we know we're looking at that. They might be looking at it, might be crossing over. So the older literature didn't necessarily have the normal controls. So they were just looking at patient variability, quite frankly, and saying, of 50 patients, it looks like PGE2 is elevated.
Yeah,
but when we
you need that within individual control, because everybody's just different across the board.
And every tumor is a little different, right? We
Yep.
know that cancer is highly heterogeneous. So when we looked at our tumors, we found that by and large PGE2 was not elevated in these tumors, and that was different from the literature. But the literature, again, older literature, I'm talking about from the 90s, when they first discovered PG2, they were using radioimmunoassays and not using these normal mucosal controls. So our paper was larger numbers, more specific, more sensitive, and we interestingly showed that PG2, PGD2, PGF2 were lower in tumor than normals. And we tried to figure out, what does that mean? And I want to put that in the context of Charles Sirhan's work, and I'll ask Ganesh to elaborate on that. But he identified something called lipid class switching. Sirhan did. And they realized that that in normal wound healing, these macrophages would change the production of pro-inflammatory lipids to pro-resolving lipid mediators in the process of normal wound healing. And that's called lipid class switching. So you might go from an LTB4 to a LXB4, which is lipoxin, which is a resolving mediator that can actually be derived from omega-6 to make it more confusing. So you can actually derive a pro-resolving lipid mediator from omega-6 arachidonic acid molecule, but that doesn't happen in colon cancer. We're not seeing that lipid class switching. So that appears to be absent, and it could be because there's this ALOX15 molecule is missing, or it could also be because of some other reason we haven't identified yet. But clearly there's a lack of lipid class switching and a lack of resolution mediators in the colon. And so we went back and looked at normal wound healing. And normal wound healing, there was an elevation of PGE2 to begin with to turn on inflammation. But there was a secondary elevation of PGE2 to turn it off. And we don't see that secondary elevation. We think we're seeing at the tumor at the time when PG2 already came on, and now it's off. Because remember, we're looking at tumors that are well established. We're not looking at carcinogenesis here. And so that bimodal PG2 peak we don't see. So we see that missing peak. So that's kind of the smoking gun for why we don't see lipid class switching. And you can't give people PG2 because it can be a problem. I'll just put it that. But maybe Ganesh can weigh in on this story.
Yeah, sure. Actually, Tim mentioned like how our approach was very comprehensive, focusing on targeted and untargeted. And there was kind of a dual purpose. One is what is in the cell wall of the tumor and healthy tissue. And that's where we use the untargeted approach using the Hokkaido University, Dr. Gowda. And what they did, they quantitated 273 lipids present in this kind of, understand the structural lipids. So what you see, we see that the lyso-PC and those which are enriched with the linoleic acid, those are high. It means that is giving the first clue itself in the tumor tissue that the molecules coming from the seed oils, those are relatively higher. That is one component. And then when we go downstream, to the very targeted. And I just want to mention the caveat that historically the lipids are in the clinical setting classified as the LDL, HDL, VLDL, just like classifying the whole world human being into the four set, very high, very short, in between or something. But here we are trying to see individual kind of lipid and what's its activity. So here again, we further laser focused on the arachidonic acid pathway because we got the clue from untargeted And then we started focusing kind of
Mhm.
down. And then I see when the team approached me, hey, can we do this 10 samples at a very first setting and see what is coming out? Kind of with surprise, my first surprise, I could see the leukotriene B4. Usually, this is a very unstable molecule. It's kind of challenging to detect in normal setting, but I could see. For the precursor of leukotriene B4, I was very confident. That is very obvious. And that's how we can start
Yeah.
connect.
And so, Ganesh, just to remind people, so basically the pathway here is omega 6s, the polyunsaturated fatty acids and seed oils.
Right.
They can then go to arachidonic acid, and then the leukotrienes are after that.
In between, there is a molecule called as the phi heat. That is the second generation, and the leukotriene B4 is the third generation. So I was very confident about the phi heat, and the phi heat is very easy to quantitate because it's relatively much more stable than the leukotriene B4. And then I saw that one, and I said, "Oh, that is very obvious, Tim, because people are eating lots of omega-6, and that's why arachidonic acid, and then there is a phi heat." But I'm kind of really curious to see more samples why this leukotriene B4 is high. Because one thing, in 1983, I believe Ben Samuelson and his colleagues received the Nobel Prize for leukotriene B4 discovery and related molecules. And that time, the resolution of inflammation field was not even born. And then I kind of see surprising, and that's why we start adding more and more samples. And then again, further, we compare to the normal tissue and the tumor tissue from the same patients. And that started making more interest. And we kind of started continuously adding more samples and expanding this project to the next level. And that we are seeing as a complete manuscript as on today, connecting directly to the processed food chain, how that chronic inflammation is there at the site of tumor.
So, I guess one of the punchlines here is, you guys are looking at colon cancer tissue, and you're seeing lipid dysregulation, and you're seeing basically more pro-inflammatory lipids. Let's just put it in very simple terms. How obviously one plausible hypothesis here as to what the origin of that is that people are eating dietary omega-6 fatty acids because of all the seed oils and everything. Are there any other plausible explanations for this? Are there alternative explanations for this? Or do we think it's very likely to be seed oil consumption?
Well, I think it could be also, omega-6s from your diet from other sources. Again, if you're eating grass-fed animals, grass-fed beef, pork, etc., that's high, 25 to 1.
You mean grain-fed?
Grain-fed, excuse me. Thank you. That's high in these ratios. Is omega-6 different from seed oil? Yes. Seed oil is a different process. We haven't talked about how it's processed, and some are highly processed, some are cold pressed, some are high oleic, low oleic. I mean, we don't really understand what all that means yet, but there are different processes for this, and probably the more processing, the worse it is. But again, we don't know for sure, but we'd suspect that. So you could get omega-6 from various components of your diet, but if you factor in, I don't eat a lot of salmon, or I do, or I don't eat a lot of nuts, or I do.
Right. Are you balancing it with the omega-3s?
Yeah, and I've heard that it's just hard to get enough omega-3s in your diet. Because of the bias of the omega-6s. And all
and if I'm understanding, so the problem isn't that people can't get enough omega-3s, period. It's really that there's so much omega-6 in the environment that you kind of have to eat an incredible amount of omega-3s.
You might say, well, let's start taking fish oil as a supplement. Well, number one, it can cause atrial fibrillation, particularly in men, if you take over, I think, 2 to 3 grams a day. So that's a risk. You don't want to get atrial fibrillation. It can cause problems with clotting and this sort of, and it hasn't been demonstrated to give great effects on heart disease and so forth. So you probably can't take enough of them to balance it. There is something called SPM, Specialized Pro-Resolving Mediators. Those are online. We're looking at those. as a potential anti-inflammatory supplement. That's what they're promoted as. I think they probably do have those effects. They're probably more potent than fish oil. Don't seem to have the AFib risk yet, but they probably have been studied in large enough populations. But again, there are things we can do probably with our diet first, but it's difficult. You might have to grow back to making your own garden in your backyard with a lot of greens and eating more nuts, walnuts, almonds, things like that, Brazil nuts, and avoiding the breads that are and salad dressing and all these other things that are loaded with the soybean oil.
And just to clarify some terminology here for people, so there's some overlap between some of these concepts. So when we say things like seed oils versus omega-6s, et cetera, so one of the keys here, I think, is that the omega-6 polyunsaturated fatty acid that's the most common in these foods in general, my understanding, is linoleic acid. And that is the precursor to arachidonic acid and a lot of the stuff you guys have been talking about.
Yeah, and what we haven't talked about is a linoleic acid can be oxidized. It can go to a lot of other things too.
Yeah.
And we don't know what all those things do and are. But arachidonic acid is certainly one pathway that they can go down.
Yeah.
It's complex. The biology is very complex.
Yeah. I mean, that's almost goes without saying, but I think one of the reasons it's so interesting, but also why there's so many different viewpoints when people start getting into this stuff, is that the biology is very complex here.
Right. And there's a lot of people trying to defend the seed oils because they're in everything. And it's going to be hard to change that. I think the market will change it, to be honest with you, just like the market's moved more companies away from sugar to artificial sugars, and stevia is now becoming more popular because it's natural and may not affect the microbiome, for example. So I think the same thing could happen with the seed oils as has happened with the sugars, moving to more natural things. It may cost a little more, but in the long run, it's going to be healthier.
Yeah, you pay a little bit more now for food, but you might pay a lot less later from your hospital bills.
Right. Or the government paying for it and your taxes and everything else. Piles up. So
there's a little bit more to discuss in the paper, but we've gotten to the essence of it. This was getting a lot of attention, which is great because I'm really interested in this field. It wasn't the field I was trained in and stuff, but I've gotten really interested in this, lipid biology and lipid dysregulation, how that ties into diet. I think it's a fascinating field, and it's really consequential stuff here. We're talking about cancer, certain cancers are on the rise, diet, whether or not, whatever your viewpoints are on diet and what is or is not healthy and all the holy wars people get into, it's obviously an important subject, and all of these things tie together in a really interesting and complex way with some of this story here. And one of the things I sort of saw people yelling different things about this, and I want to just get it directly from the guys who did the study. What's sense and what's nonsense here? There's lots of headlines out there where people are basically saying, study proves seed oils give you cancer, period. And then there's a lot of other people, some of them scientists, that were saying things like, this study had nothing to do with seed oils or diet. So where's the signal and where's the noise in those types of statements?
Yeah, it's somewhere in the middle, probably. We did not prove cause and effect of seed oils to cancer or seed oils to anything because we didn't study seed oils. We were looking at the tumor tissue in large enough sample size. No one else has done that kind of sample size in human colon tissues. And we saw what I feel is the smoking gun. So I think we have a smoking gun story where we see this inflammation that's significant in the tissues. And you've got to ask, where did it come from? It's got to come from the diet. And then you get backwards. Well, where in the diet could it possibly come from? And what's happened in the last, 50 years in our diet?
When you say it's got to come from the diet, why is that your viewpoint?
Because arachidonic acid and omega-6s these are essential fatty acids.
Body can't make them on its own.
Correct. You have to get them from the diet. So that's sort of prima facie evidence again that the diet's involved. But
so you didn't prove, when we say prove, we're being, as a scientist, we didn't prove directly that it was the seed oils, but you're basically saying it's very difficult to imagine where else this would come from.
Right, because that's been the big change.
Yeah.
And we had the same problem with cigarettes and cancer.
Right.
You remember for years, cigarettes don't cause cancer. And it would use the example that, well, not everybody gets cancer that smokes. That's true. But they get heart disease and COPD and other things and strokes and diabetes and Alzheimer's, but they're not, it's not connected causally. But we all know it actually probably is. It just took a long time to really find the exact proof and the methylation events or the mutational events.
Yeah. And we're talking about stochastic biological processes here.
Right.
Yeah.
Exactly. So this is, hard to prove that cause and effect. But I can tell you this, the dietary studies, the epidemiology studies, that's a different science than lab-based studies.
Yeah.
If you look at epidemiology and diet histories and all that, they have some inaccuracies to them, right?
Oh, yeah.
And that's why people go to meta-analyses to try to prove their case, because one study is not, the be-all and end-all of like a gene knockout study.
Yep.
Ours is also flawed because you have it's translational research. You're looking at heterogeneity of 81 patients. But there's this definite trend here that's statistically significant that we see in the lipidomics technology. And we also did spatial transcriptomics, which was looking at the gene expression of individual cells within the tissue. And we related ALOX5 and 5AP overexpression to the very tumors that had the highest LTB4 levels. So we're seeing, the enzymes responsible for breaking down the inflammatory lipids to lipid mediators are inflammatory in the actual tissues. We see the lipid levels in the tissues, and yes, we got to relate it back to something in our diet. Now, what's changed our diet? It started with margarine. It started with adding seed oils to foods instead of saturated fats. And people say, well, heart attacks have changed when we went from saturated fats to margarine. We reduced heart attack risk. Well, that may be true because you reduce saturated fats, but now if you replace them with an unbiased additive of polyunsaturated omega-6s, I think that becomes inflammatory. So, studies can be manipulated lots of different ways. That's why I like the hard science of lipidomics and tissue spatial transcriptomics and things we did, as opposed to just purely dietary histories. And I mean, how really carefully can you take dietary history on someone?
Right.
And when you go to a restaurant, right, it's Russian roulette.
Yeah, you don't know.
Well, put a Dexcom on your, or one of these glucometers on your arm, and go to a restaurant and find out how many times it spikes. There's sugar in everything. There's bad fats and everything. Although restaurants may tend to use more olive oil than processed food companies because they're cooking, right there in front of you.
Yeah, and another thing, sort of in this world of, there's a lot of complexity here. There's the biological complexity of the phenomena we're talking about, but there's also basic research, clinical research, all different types of studies that different types of scientists conduct. They all have their different strengths and weaknesses. One of the things that I want to ask you guys about, I actually asked it a little bit before, but I want to come back to it, is, you have people debating, experts debating about seed oils. Some people say, well, the seed oils are clearly pro-inflammatory because we know all of this biochemistry that's been worked out, all the stuff that you guys were talking to us about. And the story fits very beautifully in many ways, in my opinion, and in certain people's opinions. But then other people will say, they're usually more clinically oriented people. They'll say, great, that's all great. The basic biology sounds good in theory, but we don't see our randomized control trials clearly showing that seed oil consumption causes an increase in all of our favorite inflammatory markers. How do you guys think about those elements of the literature here?
I'll address it, and then Ganesh can address it. But some of these trials I've read, we did 10 weeks of increasing obesity diet for a patient and 10 weeks for a normal diet. Well, and then we looked at plasma levels of omega-6 and omega-3. And okay, is 10 weeks a lifetime? No. I mean, I don't think this is glacial stuff. It doesn't happen overnight.
Right.
It takes decades, right? So decades of exposure may cause these risks. So I think some of these studies that are randomized control, you can't do it long enough to see the effects. That's number one. Number two, I think we didn't talk about some other things that are happening, and we have no explanation for it. Why do we see an increase in colon cancer under the age of 40? There's a substantial increase in the last 10 to 20
Yeah.
years.
Yeah. And
What's that all
why in
about?
younger people? That is strange
Right.
at
So
first blush.
what has changed?
Yeah.
The only thing that has changed is really, quite frankly, diets, and maybe levels of exercise and so forth. But the children today have been exposed to more of these PUFAs, these polyunsaturated fatty acid omega 6 varieties than I was, as a
Yeah.
child growing up. And so there's a big difference. And we haven't explained it yet clinically. No one has. And we need to figure it out. But we need to, I think it's the default is we should assume that it's the diet and prove it's not.
Yeah.
As opposed to saying, oh, the diet's probably safe. Let's look for some
Yep.
other
Yep.
cause.
And I think that's
Because it's
super
something we need
important.
to do. Yeah.
Yeah. I think both things you just said are super important. So we shouldn't assume by default that these things are good unless proven otherwise, because they're so brand new. Or we don't have we don't have a long history with these things except after we started making them at an industrial scale. But Tim, something you said there I think is very important, which is like I'm speaking from my perspective here, so I'm not going to speak for
Right.
anyone else. So just for everyone listening, no one is saying randomized control trials aren't great and we shouldn't look for things in them. But a key limitation with this area of biology is, as you said, Tim, a lot of the stuff moves at a slow pace in terms of, our diets shift lipid profiles and things in ways that won't manifest in clinically relevant markers that we can measure unless you're talking about months or even years of time. And just to put a little bit more meat on the bone there are studies in animals, and sometimes people say, yeah, it's an animal study, it's not a human study, we should ignore it. But there are animal studies that very clearly show that when you change the dietary fatty acid composition in rodents, you can see substantial changes, but they take sometimes 16 weeks to manifest in a mouse. And that would be like 10 plus years in a human being. And as you said, Tim, a lot of these clinical trials, even when they're well conducted, and even though a 10-week long clinical trial is a big, expensive effort, and that's very commendable. But unfortunately, a lot of this biology would require us to do very, very, very long studies
Well,
that really just can't be done.
A number of the older studies, the Framingham study, the Nurses' Health study, those followed nurses and people over years, decades. And granted, diet histories or diet recall histories are not that great because what did you eat for breakfast 3 days ago? Well, if you didn't fill out your diet right away, you may not remember, right? So how accurate is that is another question. But those studies are what it takes to that's why those studies identified LDL and risk of heart attack with high LDL levels. They looked over years at people, not just a randomized control trial. So I think there are artifacts and there are weaknesses to any study. A lot more works. And the other part of this whole thing, again, is this microbiome picture. So again, I challenge you to take a loaf of bread off the shelf and look at everything that could be in it.
Yeah.
And soybean oil is one of the things, but there's often other things in it like preservatives and natural flavors and dyes and this and that. What do they do to the microbiome? I don't know. I mean, it's WNL. We never looked, and because we haven't looked yet. We didn't have the technology, but now we do. And we now know that the microbiome controls the immunity, and there's the gut-brain axis. There's some evidence of the gut-heart axis. So if the gut microbiome is so critical, and the food, and obviously it makes sense, right? It's the food is nourishing our entire body. Got to go through that gut passage first. It's kind of like common sense. So I would say the common sense approach is to use the default that these things are likely problematic, prove that they're not, and be much more, or take them out of the diet and just substitute. I think the American public is going to, eventually have an outcry and force the food industry to change, and they will. They respond to the market. If people stop buying goods, the market responds. That's one of the beauties of capitalism. They will change. So.
Ganesh, anything to add to that in terms of how, like, as a hardcore lipidomics guy, how you sort of integrate and interpret the clinical versus the basic research literature on this stuff?
Sure. Yeah. And this applies to kind of multiple formulations or products in the market. What is assumption? And I know when I talk to my 15-year-old son, a high schooler, and he said, hey, and he said, read the package. Oh, this is real. This is green. This is healthy. And that's where perception, but he don't read like, okay, how much the content of these seed oils is there and what is the impact on the body. And that's what the Americans did. They are doing like, oh, this is written as a healthy, real, natural, and kind of it's hijacking your senses, and you are following that 100% and using those products. But when you see this kind of real versus unreal study, or how much we have to believe, and that happened to the omega-3 fatty acids. And that's true. Some people say, hey, omega-3 is really helping for me, for my kind of cardiac stuff. And some 50% saying, oh, no, it's a garbage, don't use it. And then the question is, hey, is your body's enzyme to process omega-3 is working? Oh, I don't know. And that's the kind of reaction of the kind of in the population study. But here we are talking about totally different approach. Hey, what are the levels of these particular very bioactive lipids at the site of where there is a problem, like a tumor? And that's where the chemistry clearly telling us that, hey, these are from. Now the question, hey, tell me how you can prove it. Because one thing we have to believe very kind of old-fashioned study, and that's where this coin essential fats. Those were husband and wife, the George Bird and the Mildred Bird. What they did, they kind of removed totally fat from the rat diet. What they noticed, those rats were dying way earlier. They have skin problems and many problems. And as soon as they add 2-3 drops of the fat, and then they start surviving and much more better health. It means the fat is essential.
Mm-hmm.
One confounding factors we have to understand, even the glucose also disrupt the lipid metabolism. Fructose also disrupt the lipid metabolism. So many dietary kind of choices, the impact on lipid metabolism, but this was never studied. So that is the one reason, yes, there is possible everyone can see, oh, there is a lot of smoke in the study because we are not considering what the populations were eating. That's 100% right. But we know that these molecules are exclusively come from only fat that every day we eat. And that's a very obvious connection for us, how it is kind of changing the precision of our immune system, which is key for our health.
I was just reading some food labels from bread.
Yeah.
I went to the store and I'll just read you one out loud. You'd think in bread, you need water and flour. Actually, it's all you need, and maybe some yeast, right? But here's just one, this typical loaf of bread: water, modified wheat starch. Well, why is it modified? How did they modify it? Wheat protein isolate. Okay, why not just wheat and flour? The sunflower flour, vegetable oil, soybean, preservatives, including calcium propionate, sorbic acid, monoglycerides, soy lecithin, guar gum, xanthan gum, maltodextrin, citric acid. Why do you need all those things? I'm not sure why you do. Some are, anti-mold, some are anti-this. What are they doing to the microbiome?
Right.
So if we all went back to make our own bread, or, growing our own vegetables and eating right, I think it would be a big difference in health.
So let me ask you a related question. So that sounds great in theory, but the fact is most people can't make their own bread. They couldn't even, if someone lives in a city or they live in one of these food deserts, like, going back to an ancestral diet just may not be in the cards for most people. How do you guys, I'm guessing you guys aren't farmers on the side while you're doing your full-time science work here. What do you guys do to help avoid consuming so much of this stuff in your own life?
Yeah, so what I did, I read a book called Square Foot Gardening, and anybody can do this. You can get a ton of vegetables and things out of a small area of a small plot that's raised in your backyard. That's number one. And I can tell you, with kale seeds, you can grow a ton of kale. It's amazing, actually, with what little effort. Bread, there's 1,000 videos online now, right? Anywhere, how to make bread. It's so simple, right? So you could do that in your house. I think if we went back to that, like our grandmothers did, or your great grandmother, you would see that life was different. It's much more convenient now to get a packaged food out. It's preserved. Preservation, health in the US improved with refrigeration. When we started, when refrigeration came along, we preserved foods without spoilage and people didn't get sick. But then we went to dry preservation. You notice when NASA sends their astronauts up, they're not preserved, they're freeze-dried foods, right? And so if we went to freeze drying or we went to other approaches of eating fresher foods, going to a local bakery and get it right there. If you go to other countries, if you go around the corner in Italy or France or these other European countries, Portugal, you can find bakeries everywhere, right? And you can find these things. We just don't have that here. We're in this fast food mentality of packaged foods, and they're ultra processing associated with that, which we haven't gotten into. It's just there's a whole, other aberration of food that's related to that process.
Mm-hmm.
I just want to add, like in the kind of at my end, life's ABCD is very simple. What do you mean by BCD is birth and death? There is a C, and that C is a choice. The choice actually determines the distance between the B and D. That's a birth and death. If your choice is conscious and you're really understanding that, okay, what things I'm buying, what kind of things I'm putting in mouth, and what's the impact on the health, and then that will increase the distance. But if I don't care, there is I'll just buy it because I'm genetically okay. And if we are taking that concept and the environment is not impacting health, then definitely that the distance between the B and D is going to reduce. So that's how. So I prefer personally to use, if we have a time, then the first thing is use a home cooked kind of meal as much as possible. And if you are buying the product, make sure even it is written as real or natural, not necessarily it is natural or real or with the healthy stuff, make your call that, okay, and what's your lifestyle? At the same time, you have to be active and taking the enough sleep, because those are the three components. That activity and the sleep is not really very well studied in the context of the lipid metabolism and the cancer, but we are kind of starting snapshot and understanding, okay, this is the one case. But in terms of the health, I take personally the simple theory of to kind of increase the distance between the B and D. The best is kind of give very high weight on the C is what I want to choose or what is my choice.
Mm-hmm. Mm-hmm. And so I guess the bottom line, bird's eye view, but bottom line on seed oils for you guys. I mean, do you come down clearly on the side of seed oil consumption should be minimized because you think the evidence is compelling enough to really warrant that conclusion?
I actually do. I think you should start looking at your labels and realize that how much seed oil you have on your shelves and how much is going into your mouth. So if you go to Super Bowl Sunday, you're gonna get a boatload of seed oils, right? From the chicken wings, with not seed oil, but from omega-6s, let's say, from the chicken wings to the chips to the dips, just loaded with seed oils. And so try to avoid. It's hard to find bread. Actually, you can find bread. And I'll give a plug for a store. I went to a store the other day, Ingles, and they had actually a number of breads that did not have soybean oil on them, but they were all freshly baked there. And I've seen some at our local store here that uses olive oil. Crackers, when you buy crackers, it's very hard to find crackers that aren't loaded with, some kind of seed oil. But you can find. They're often more expensive. But that's another choice. Pies, cake, cookies, same thing. You have to look at all that, cereals, all those things. So just start making label choices, reading the labels. There's a label app that I found. It's called Bobby Approved. It's free, BOBBY approved, and it scans with your phone the label. So you can just scan any label. I think it's the barcode on the label has all the information, the food label, and it will say Bobby approved or not. And often the disapproval comes because there's a seed oil in the product. So it's actually very easy to find it because you can't read these labels sometimes. They're so small.
Right.
But you scan it with this web app or phone app. You can again, it's free to download. So I would suggest something like that.
Interesting.
I just want to add, and I 100% agree with the Tim that it's time to limit the kind of oils, with seed oils, especially the pro-inflammatory. But another component that we really don't know and that we have to be cautious, one, when these oils are heated, for the fried foods, and that's kind of use many fast food chain restaurants. Because, and how frequently they are changing the oils. Is it the 1 hour oil heating is causing what kind of molecules, and if you are eating the food? So this is kind of whole totally different question at this point of time, but still that is very understudied, and that's why kind of we have to be very cautious that okay, how these oils are used in a different industry and to what extent, and when we are choosing the food, and then what type of food, so that we can keep the whole metabolism in a healthy side, and that will kind of help our immune system to stay in a precise mode towards the precision of the health.
Mm-hmm. Yeah, heating oils can lead to trans fats and oxidized fats that can have problems. And I think McDonald's, for example, cooks its fries in canola oil, corn oil, and soybean oil. And when you heat those up to a certain temperature, you can get oxidation of lipids and problems. So, and also the process of like making certain oils like canola is it's heated to high heats to make it. I tend to use olive oil and avocado oil and something called algae oil to cook with, because they have lower smoke temperatures. You don't want to smoke oils. Once you start smoking, you're oxidizing them. So whenever you're cooking, you want to keep it low. It's low temperature cooking is a problem. It's not a problem.
Well, we've covered a lot, and one last thing I want to ask you guys. Your study was looking at colon cancer, but by the nature of this stuff, how these lipid mediators are created, the connection with inflammation. There's no right. If I'm reading all this stuff right, there's no reason to think that this stuff applies only to colon cancer. This stuff probably ties into, I mean, in theory, any inflammation-based disease out there, which is, I mean, a lot of stuff here. So this has sort of general implications for health in a very general way, doesn't it?
Certainly, I'll speak to other cancers. We started looking at some other tumors, and they're definitely inflamed. So Ganesh, maybe you could talk a bit about heart disease and this inflammation story, because he's a cardiology researcher to begin with.
Yeah. Sure. Actually, we are studying last 10, 15 years, the omega-3 and omega-6, with more focus on how these fatty acids get used, what type of enzymes. So that's our key focus. And then what we are seeing, like, okay, overall health is depend on the intake of those fatty acids. But what they are doing, the molecules coming from those fat actually helps to guide our immune cells and the inflammation. Now, historically, as we discussed, the inflammation is helpful and that is necessary, but only when it is at right time activated, and then it is controlled within a certain time, very precisely, with the help of our immune cells. Those immune cells are nothing but the firefighters. And those firefighters, again, get out from that location. We come to the normal site. That's the healthy. So overall inflammation field is kind of continuously going in the growth mode, and we are saying, but now we are suddenly started saying, oh, there is a growth of chronic inflammation, and we have a big confusion between, hey, what is the inflammation? You're saying acute is good and chronic is not good. I confuse, but here is the key. But this all is coming from that 1% of the immune cells that got miscalibrated with the fatty acids or the fat which is present in that seed oils or other processed food. And that's how we have a kind of continuous growth of chronic inflammation. We have studied at a multiple level, just adding the excess fat in the animal diet, and then how these enzymes, mediators, and receptor change. We work at the very immune specific enzyme level, taking out this particular enzyme from one cell type, like a macrophages, as Tim mentioned, and then what is the impact. And then also we studied by giving one single individual lipid mediators to the mice after the heart attack, and then we see there is a better cardiac repair if these molecules is coming from the healthy fat like omega-3, and that's the specialized pro-resolving mediators, especially the resolving D1. One most surprising thing that when you take the diet, then the first step is a diet, second enzyme, third lipid mediators, and the last in the linear fashion is the receptor. When the receptor is not there, means that last step, the mice, one particular type of receptor that's called as a formyl peptide receptor, which is necessary for resolving D1. And when that receptor is absent, the mice become obese on normal diet. We don't need to feed them any high fat or high omega-6 diet, normal diet, and they die within 6 months to 1 year. Means suggesting that, okay, our receptors are totally contributing to the whole health. If just the receptor is a dysfunction, then some people may become the obese that we don't know as on today. Means conveying the message that our whole immune system or immune function is depending on those 4 components of the fat biology. So overall, the fat has not only the role in the cancer or one particular type of disease, but the whole health, whole body health. The total health is depend on these 4 components and the alignment with the immune cells, that the leukocytes.
Mm-hmm, mm-hmm. Well, I want to thank you guys for your time. We covered a lot today. I'll let you guys decide if there's any final thoughts you want to leave people with, but it sounds like we have more to look forward to, and that these imbalances and lipid dysregulation that leads to inflammatory states, it sounds like we have more research to look forward to that's going to show how this stuff ties into other diseases, whether it's cancer or other forms of disease.
Yeah, and I would say all that's correct, and people should focus on diet, exercise, and sleep as our Dr. Halade suggested, and start reading labels and trying to reduce your intake of some of these things. Grow a garden.
I just mentioned that this is just the beginnings for understanding because of availability of the technology. As the technology is going to grow, and then we will have more understanding how these lipids are functioning for various parts of the organ and how they are helping to control our immune system in the right direction.
All right. Well, Professors Tim Yeatman, Ganesh Halade, thank you very much for your time. And yeah, I really enjoyed looking at this at study. Really cool stuff. Lipidomics. The technology is really interesting, and I like what you guys did. So I look forward to following your research in the coming months and years.
Thank you so much for having us.
Thank you very much.
Hey everyone, I want to share with you a new AI-powered research tool I've been using to find scientific papers and learn from the literature. It's called Consensus. It is an AI-powered search tool built using large language models trained to help you find and understand the best science faster. Consensus helps you not only find individual papers, but its AI models will help you assess research quality and even synthesize insights across multiple papers for you. You can ask it general questions about any topic you might have, such as what are the known risks and benefits of a certain prescription drug? What do we know about how a certain food or nutrient affects the human body? Anything you can think of. Its pro analysis feature will actually provide you with a concise written summary of what the literature says, broken down point by point, highlighting the strength of the evidence across papers and providing you with individual studies ranked by the relevance and including additional information like citation rates, journal quality, and more. You can also save, cite, and share individual studies with a single click. Unlike other AI and search tools like ChatGPT, PubMed, or Google Scholar, Consensus uses over 20 custom large language models that are developed and trained for this specific purpose. The base Consensus product is totally free, but the premium product that you can subscribe to has better features and unlimited queries. I highly recommend this if you are using outdated tools like PubMed or Google Scholar, or you just do your own research. For a limited time, you can use the code MINDMATTERSPECIAL, all one word, to get a free premium subscription for 1 year. That is a $150 value for free if you use the code MINDMATTERSPECIAL today. So click the link in the episode description or check my support page and give Consensus a try. Use the code MINDMATTERSPECIAL to unlock your free premium subscription today.